Good morning, guys. As promised, I am going to kind of fill in the blanks with genetics for the pieces that I feel you guys are having trouble with. And so since I'm going to be gone today, I figured I would still be here in spirit and on the computer screen. So looking at these notes and going over these kind of in a fast and furious manner, this first page anyway, to kind of recap what we've talked about in class. The study of heredity is genetics, and so it's nothing more that's really all that fabulous, but genetics, this is going to be interesting. You got to bear with me. I'm going to change my pen size. So the study of heredity and how things are passed down are through genetics, and specific characteristics that we can look at. The example here given us seed color, or we can look at hair color, or we can look at gender, or we can look at skin color, what color eyes we have, all those different types of characteristics we call traits. So we're going to go there. That's a little better. It's still messy, but you get the point. And when we look at the chemical factors of those traits, so the DNA pieces, the itty-bitty parts that control the traits, we're going to call those the genes. So the genes that can control the traits are the DNA pieces. So when we looked at a segment of DNA and we coded the amino acids to form the polypeptide out of the strand, that was a gene. So we can do that for hair color, we can do that for eye color, we can do it for all the traits that we have. Some of the gene segments are shorter and some of them are bigger. It just depends on what it is. But the chemical pieces, the DNA pieces that we can look at that control the traits, we call genes. And then when we are talking about a gene, we can talk about those as big T and little t. Not that they're really capital and lowercase, it's just that we use that as a model. And so when we do that, we call those alleles. I can use any letter of the alphabet. It doesn't matter. What matters is that are we going to represent it as being capital or lowercase. So that's really the difference. And we'll talk about that in just a minute. But when we talk about genes, and I say big T, little t, or big G, little g, I'm going to be talking about the alleles. And that's what they're called. We use them as a model. We've spent a lot of time on this in meiosis, but when we are looking at how things are passed down from mom and dad, or passed down from generation to generation, we have to look at that passing of genetic material through the sex cells. So whether it's the sperm or it's the egg, that's how traits are passed down. And so we know those as gametes, and we've already talked about that. Okay, so on the first screencast-o-matic that I did, a lot of time was spent on this. And so it was the father of genetics, which was Gregor Mendel. And Gregor Mendel concluded a few different things from his study. Remember, he's the guy that studied the pea plants, and he's considered the father of genetics. I'm sorry, this looks so horrible. But he, throughout his studies, which it took quite a while, even though pea plants are really easy to manipulate because we can pollinate them and cross-pollinate them pretty easy. One of the things that he concluded from his studies is that inheritance is controlled by factors passed on from one generation to the next. For us, that's a really moot point. We know that inheritance is controlled and it's passed down from generation to generation. But when Gregor Mendel was doing his studies, that wasn't common practice. That wasn't common knowledge. And so that was one of the things that he concluded. The number 2, the other things that he concluded through his studies is that some alleles are dominant and some are recessive. And so some are capital and some are lowercase. So when we talk about dominant and recessive characteristics or alleles, we're going to talk about them as being capital and lowercase, or dominant and recessive. So some alleles are dominant. Dominant should go here. Let me see how good I can fit this in. Not very well. Just bear with me. I'll have to find a better way to do this. So some alleles are dominant and the others are recessive. Ew. This doesn't really work very well for a left-handed person either. Okay, so the third thing that we talked about in meiosis is the segregation. So the alleles are separated when the formation of the egg or the sperm. So think about that really long process that I had you guys kind of work through and manipulate with the pictures for meiosis, and looking at the colors of chromosomes and how they switched information, and how is it that we started with 1 and we ended up with 4 different cells at the end, and they're not even complete cells; they're just egg cells and sperm cells. And so knowing that the father and the mother give half of their genetic information, and the half of the genetic information that they give is not the same from egg cell to egg cell, and it's not the same from sperm cell to sperm cell. So that segregation is what causes lots of different type of variability with gene expression. So number 3 is going to be segregation. I'm going to have to blow this up. Seg. G. R. E. It's not helping. I'm having to put my pinky down to write segregate. Segregation. All right, and the 4th thing that he concluded with his genetic study with pea plants is independent assortment. And so the genes that form the different traits are going to go independently, no rhyme or reason, as to where maybe the blue hair gene, or not blue hair, blue eye gene that mom's going to pass on is going to go to one egg cell, as to where maybe the hazel eye gene is going to go to another egg cell. And so independent assortment lets us know that the genetics of genes that are found in each sperm cell or in each egg cell, during the formation of those, they are going to split independently of one another. That you have kind of, think about it, if you have 2 copies, and if we're making a sperm cell, then one copy is going to go to one copy is going to go to another. And so that's how you can kind of think about that. So that's independent assortment for number 4. How in the world am I going to fit this on here? Independent assortment. Sorry. That's Assortment. Blah, blah, blah. I'm getting tongue-tied. Okay, so the itty-bitty review for that one. So let's get down to some more nitty-gritty, and these things that you guys should have picked up on during your PBL. But if not, this is where I'm going to clarify for you. So when we have particular characteristics that occur in the phenotype if one allele is present. So, 1. Phenotype is a physical characteristic. It's actually what we can see. Like, you can see that I have red hair. And you can see maybe I have some freckles on my nose. I used to have a lot more. you can see your hair color, your skin color, all types of different characteristics. So if I'm talking about a phenotype, it's something that is observable. Even like with your blood types, your phenotype, if I say you have A blood, B blood, O blood, those are observable characteristics. Even though we have to do a little something extra to figure it out, it's still observable. So when it occurs in the phenotype, if I can see it, if one allele, so one letter is present, then we're going to call that dominant. Okay, it's going to be dominant. Dominant. Okay. Dominant. Guess what the other one is? It's the opposite. So, if that occurs in the phenotype, if one allele is present, then down here, it must be homozygous to occur in the phenotype. Maybe, I wonder if I could do, I could. So, what is homozygous? Homozygous means the same, and when we have alleles that are the same, like these little guys you see down here, big T, little t, or little t, those are called homozygous. Homozygous means same. And so when alleles are the same, meaning the same capital or the same lower, we're going to call that homozygous. So, let me do that one first, and then the one above that will make a little bit more sense. All right. So, if we're looking at the difference between dominant and recessive, when we have recessive, so lowercase letters, we need two of them to be the same in order for that trait to come out. So, let's think big T, little t is tall and short. Okay, if I have two big T's, and big T stands for tall, then that person could be tall, or that plant, or whatever. It's actually more complicated than that, but we're just going to go with it. So if I have something that is two capital letters, it's homozygous. If I have something that's two little letters, then it is homozygous. But in order for the little letters to show up, there has to be two of them. If there's a capital one, the capital one's going to dominate over the little one, and that is not going to show up. So down here, I'm going to write recessive. Okay. Recessive must be homozygous to occur in the phenotype. So it must be two little t's in order to show it. If there's a big T with it, it's going to overpower. So if I'm doing the big T, little t, tall, if I have two big T's, that's a tall person. If I have a big T, little t, that's a tall person because the big T overpowers the little t. But if I have two little t's, two recessives, then that is going to give us a short person. And if I have two different ones, so the big T, little t, rather than homozygous, that is called heterozygous when it's different. So that is heterozygous. And I will start with review on that real quick and then do the other part of these notes in the next video.